Well, hi again, Mr. Bleeker here, and welcome back. In lesson number 3, we're going to focus in on what happens when an action potential gets to the end of the axon terminals and reaches a little junction called the synaptic cleft. How does it continue this chemical reaction and produce an action potential in the next or adjacent nerve cell? Really, what we have to get is a signal that goes from axon and then reinitiates the signal at the dendrites of the next nerve. Okay, some pretty important things for you to know in this chapter. And if you basically click on lesson 3, it'll pop this up. And what we're going to look at is what happens when we get to the end there and we reach this right here. This is a little bit of space called the synaptic cleft, and this is the end of the axon. So this will all make sense soon, I promise. But it's really important that you understand how a nerve impulse is transmitted between neurons. That's the whole name of the game. So let's go here. So let's say that you decide to put on your coat, for example, and you're going to send signals down to your muscles to move. Okay. So the signal's generated in the brain. We'll call this conscious or voluntary control. And that signal will travel from the cell. Okay, here's your dendrites, obviously, but we'll call this the originating cell. And it'll go down basically from nerve cell to nerve cell. So you need to know your dendrites, your cell body, which is the main part of the cell, right? This is known as the soma down here, cell body, another word for it. The axon, in this case, is myelinated, and we know why, because we want the signal to jump from node of Ranvier to node of Ranvier. Now, when you look at a couple of nerve cells stacked, you're aware of how the action potential works, and we get to this region right here. So we have to define the one side from the other. So we say if we're at the end of the axon here, we come to the end, this is the presynaptic neuron, because this is the synapse, right? So this is pre, and makes sense, this is post. And as the action potential's coming along, there'll be the depolarization happening all along the membrane. And then you get to the end, and you'll notice some very interesting chemistry here. And what you have some little, they're called little channels. Now, if you've been paying attention, this whole what we've been looking at has all been a little bit of a chemical game. I need to pop out to the notes for just 1 second. There's a few things we need to write down. Okay, just my notes are hiding. Okay, just turn reflection back on here. Airplay my notes. There we go. Okay, so now that we've come down, the signal has jumped from node of Ranvier to node of Ranvier, so to speak. And we reached here, unmyelinated. We get out to the axon bulbs right there. And we need to send the signal to the next nerve. Or, alternatively, we could be sending a signal to a muscle. Like, ultimately, you will get down to a little junction between the nerve and the muscle. So that right there, we'll look at when all is said and done, we'll say, well, what if a muscle is the final target? Well, we'll get there. Let's just look at from nerve cell to nerve cell for now. Okay, so this is a nice, really nice drawing here. The nerve impulse comes along, and of course we've got our exchange of our ions, right, where it was, oops, gotta hold that straight. There we go. Where we had this exchange of sodium to the inside. So if the nerve impulse is here, right, let's say it's achieved this point, this is our depolarization of the membrane. I'll just draw this here. Now, you can think of this chemical reactions occurring, and when it hits the end, we hit these little channels. Now here comes the secret, and there's lots of calcium channels. Just draw two. Outside of the membrane, you have a lot of calcium on hand, and that calcium can flow in through channels if the channels open up. And that's exactly what happens. So calcium ions will flow in, and they have a little bit of a different effect now. What they do is they cause a rush or a release of, oops, these little guys down here, these vesicles. Now these vesicles contain neurotransmitters, and you've probably heard of those. For example, if you take certain substances, you can cause increased sort of rates of nerve firing. Speed will cause incredible amounts of neurotransmitters to be released. So your action potentials will go across very quickly across the synapses. If you consume alcohol, you can inhibit the production of these neurotransmitters. And we're actually going to look at a couple examples of those. But once these neurotransmitters are released like this into the cleft, right there, basically this hollow space, they open little doors on the other side. So this is kind of interesting. The neurotransmitters are released when the nerve impulse reaches the end. So it's like a little calcium nudge, and these vesicles form. They release the neurotransmitters, and think of the neurotransmitters as like little doormen, because as they go to this side, let's zoom in on this, it's worth it. As they go across the synaptic cleft, they open. You can see in this case, this one is closed. You're not getting across there. That's because nobody opened the door. Now, who opens the door is the neurotransmitter. This neurotransmitter right here has opened the door. So what can go through? Now, don't say calcium. I mean, I've had students say that before. No, we're not talking about calcium anymore. Calcium got all this going. Calcium causes the release of the neurotransmitter substances. What forms an action potential in the first place? Let me think about that. Oh, you mean the sudden rush of sodium ions in and potassium, how they sort of flee to the outside? That reversal of polarity across the membrane? That's exactly what happens. So the ion channels open, sodium flows in, right? You get that massive flow of sodium in. You get a little tiny flow of potassium in the other direction. Not a big deal, but that causes the action potential now to form. Lame lightning bolt, I know. So I'll fill it in as if that makes it better. And your action potential, your AP, carries on just as it did before. Whoa, back up. Let's take a look at that. Let's take a look at this in a little more detail. Okay. We can say that these are voltage-gated calcium channels. Think of the nerve impulse as a bioelectric impulse traveling along the membrane, because that's exactly what it is. And just like your dishwasher, this is just a little different thing that flies open. I mean, last time we talked about sodium as being like the soap dispenser. Well, maybe this is the one that lets another dispenser that lets maybe some some water softening go into basically into circulation. But in this case, what's being thrown open is a little channel that lets calcium in. And it's calcium that just causes the whole ball rolling, because once calcium floods in, then the synaptic vesicles there are going to let a whole bunch of neurotransmitters head towards the junction. So it's a sudden rush. And that's good, because you want a sudden rush because you want to get that signal started on the postsynaptic. Notice I said post. You want that signal started on the postsynaptic membrane big time. Because if you don't, well, then the signal will die. Okay, so before we get into signal blockers and all that yummy stuff, just fun in and of itself, there we go, I'd like to look at this in a little more detail. So here we go. There, now you know these are the calcium channels here waiting. So presynaptic, jump the cliff here with neurotransmitters, postsynaptic. So here comes your signal, right, at the axonal hillock there. not showing the saltatory jumps, but that's okay. And there's calcium waiting. And stop. Oh, shoot. It was so good there for a minute. That's okay. It's just as nice twice. I guess I should have just hit pause. But this would have been perfect if we'd seen this little saltatory jump. Okay. And here we go. So there's the impulse that throws open the calcium gates, and the sudden rush of calcium initiates the release of our neurotransmitters. Now, you might not have really been paying attention to it, but it causes a little bit of a chemical change, and these now will head for the synaptic cleft and release, um, uh, basically release the neurotransmitters over here and open up these gates. So here come the doormen. Now, I should stop and pause for 1 second to say there are different kinds of neurotransmitters. You have some that are excitatory, like acetylcholine, and you have some that are inhibitory. Now, I'll show you a list of those. We mention a few. We don't expect you to memorize massive lists of them. There's so many. It's kind of unbelievable. But let's see this through to the conclusion here. Okay. So our little neurotransmitter doormen come over here and either open or hold the doors closed. These little green balls here are sodium. They can get a signal started. There we go. Now you see how those doors have opened up? Well, they're wide enough to allow the sodium rush that we need for another action potential. I'll give it a moment and we'll let our sodium rush in. And there's our action potential, which started on the postsynaptic side of the neuron. And that signal will, of course, go from the dendrites. This is pre-dendritic. Ooh, pause. I want to see that. This is pre-dendritic, of course. And you'll kind of go from these little, just down here. Think of this as the dendrite tips. And then you'll go down to the dendrites, to the cell body, and then, of course, to the axon on the other side. Now, you'll notice the doormen go back. They're actually reabsorbed. Neurotransmitters are reabsorbed because they've got a repetitive job. So this is really smart by our bodies. This is also a moment where you look at it and you say, what if we messed with the neurotransmitters? What could possibly happen? Well, there's a lot of different kind of neurotransmitters. And that's something we could do a focus on right now. So I can't help myself. I have to show you this. And this is on the course site. And there's a lot of things that can happen. Different things that we take can affect the release of our neurotransmitters. Some of the things that we consume can mess with things in the cleft. And some things can not only mess with things in the cleft, but they can plug these gates on the other side. So let's look at a few. So currently alcohol is loaded. Now over here what I'll say is alcohol, on the left if we look at normal transmission, right? Here's our neurotransmitter horde going into the synaptic cleft, and you can see these are normal firing rates. So we're getting enough sodium through to have a pretty good action potential. Look on the alcohol side. What happens is alcohol, represented with these little green things, is affecting this here. So we're not getting sodium in there because alcohol is acting as a blocker. Not only that, alcohol is also messing up here with the release of our neurotransmitters. So now when you look at this represents activity, you see the normal firing rate is doing great here. And on the alcohol side, it's slowed right down. A crazy example of this is to watch somebody throw their hands out as if they're boxing. Their hand speed when it's normal will be tremendously fast or fast enough, and shadow boxing or play boxing, absolutely you could see the effect on the muscles where they just can't fire at the same rate and it's just completely slow, right? You don't want to box when you've got inhibitors in you, and that's exactly what you're seeing here. Now caffeine is a stimulant. We feel the jolt, but it actually slows down the firing rate of or the release rate of our neurotransmitters and such. What's going on? Well, here's caffeine, and it's messing like crazy in the synaptic cleft. And uh, if you have a really high rate of firing, if I remember one time I went to I watched Cirque du Soleil. I was watching Con. It so much firing. I was taking in so much it almost made me feel sleepy, right? But caffeine can slow down that. At the end of the day, we have a really sort of high firing rate, and I felt that when I was watching just all that action at Cirque, it actually made me feel extraordinarily tired. Caffeine can inhibit this active firing rate, and here it is, and so it'll make it feel like we're not sleepy. So it's inhibiting one neurotransmitter called adenosine. There's several, and if it does that, it will slow down your firing rate, and it's actually kind of inhibitory in that way. Nicotine is kind of interesting when you think about neurotransmission because it causes more action potentials. And let's just look at the activity. So here's our normal activity here, and think of this as throwing open the gates all the like much faster, and you're seeing the neurotransmitters being released much more. So you're getting many more impulses here, right? And one of the side effects you think about nicotine is it when you're smoking, people who try to quit will get weight gain, and this takes energy here. This in the brain, like you think of the calories, 2/5 of your calories are used just thinking and doing nervous processes. Well, you can see there's a lot more going on here, so this require a little bit more energy. So yes, your nervous functions do consume energy, and this will also affect your muscular function as well. So that'll burn more energy. Heroin, if we take a little peek at this, itself has a huge increase in the firing rate. And when you these drugs, it's a little bit difficult with heroin, but what we have observed is we see that the synaptic rate is 7 times what it should normally be. And what we're seeing is a tremendous release of neurotransmitters. It's a lot faster. And the neurotransmitters, of course, they're being reabsorbed really quickly here. So on heroin, if you increase the impulses like this, when you look at the effects this can have on the brain, you can be perceiving things that you don't normally perceive. Definitely, we're getting a race of synaptic activity. Cocaine here with the sense of euphoria. Again, we're seeing an increase in activity, and there it is messing right in the synaptic cleft. Some things can affect things by blocking, and if you cocaine itself, if you get an issue with the uptake of one neurotransmitter called dopamine, and that's not good. Dopamine for your brain is like a chemical reset. So some of these substances can not only increase your firing rate, some can affect uptake. If you don't let these guys go back, if you leave your neurotransmitters in the cleft, you could see the buzz of activity here. The reason why there's a buzz, it's not like it's coming pretty fast here, but this is being interfered with. So of course that causes more activity because the neurotransmitters can't get out of the way, and they do what neurotransmitters do. They keep throwing open the doors. Last one I wanted to chat about was depression. Now, depression, not as many neurotransmitters are being released, so there is chemical treatment for depression, but you got to be pretty careful with your doses, right? You don't want to interfere with sleep cycles either. But we need to get the amount of neurotransmitter sort of up to snuff here. It's not firing as much as it should, right? You can see the healthy side is a buzz of activity, and over here, not so fast. So in some cases, you're not getting as much released, right? Or perhaps you're getting a bit more speed on the uptake, right? And these dormant don't get a chance to open and let the sodium ions through as fast as should normally occur. And you can see the under in depression, these it's 60% less as far as postsynaptic firing rate, and you can sort of see why here. Just not getting the signal that it should. Very neat animation. Really like that one. The last thing that I wanted to say, kind of before we're finished here, is I wanted to look at the neuromuscular junction. So if a motor nerve tells a muscle to do something, what does that look like? Like we've been looking at nerves connecting to nerves with synapses. But what if we're not talking about nerves anymore? Now these are muscle fibers, these little bundles. So as the impulse arrives from uh, this is your spinal column here, and this is called a dorsal nerve root. So when that synapse instead comes to a muscle, now we're looking at basically muscular reception of the action potential. And it's really not all that different. Here's your synaptic vesicles, here's your postsynaptic side. And I really like this animation for one reason. Why? Pause. There is your neurotransmitter. Now, if you see what your neurotransmitter does, is this is how it opens the door. It's a lock and key kind of fit. When it fits in here, this channel opens up and your sodium ions can rush through the membrane. Let that continue. And as they do, this is how action potentials do their thing, right? You get the action potential firing on the other side of the membrane. Now, when that action potential kicks in, this will release chemicals to the muscle, and the muscle cells can contract. We don't really need to go much more into detail. We used to talk about muscular contraction. We looked at calcium and sodium and things like that. Maybe it might come back to the curriculum. It used to be in Biology 12 back in the good old days, like when I went to school, circa 1989. Yes, that dates me a little bit, but I wanted you to see it here. Okay, so just a few more things. Let's make sure we have sort of our final um, notes in place. I always like to make sure I didn't miss one last thing. And I'll get my reflection going. There we are. Signal blockers. I almost forgot about curare. Good thing I came back. Curare is basically like a little poison that would be put on darts or on arrows used by some tribes in the Amazon, and it stops nervous transmission. So if you stop nervous transmission, especially to the heart and lungs, that's it. That's game over. The black widow spider venom itself is really toxic because it causes almost bursting in the synapses. There's so much activity that this synapse becomes malformed. It just becomes huge, and if it's stretched and distorted, that's game over. If your synapses are damaged, there's no real coming back from them. So I just wanted to reiterate the importance of those little calcium channels, right, and how they get thrown open. That's a really big thing in getting the neurotransmitters to rush across the synaptic cleft. And I think I said that twice for some odd reason. There's different kinds of neurotransmitters, and neurotransmitters are a lot like hormones. Acetylcholine is itself excitatory, right? Especially on muscles. If you remember, I did a lab where we put acetylcholine on heart tissue in it. You see the rate increase. Acetylcholine is excitatory. Some of these hormones are made sort of protein based. You'll notice right here. I'll just do a little highlighteroo. They say they're amine based, and you know that group. That's an amino group. This one, dopamine, gives you a sense of euphoria, a sense of well-being, right? It's a nice reset hormone. When you sleep, serotonin, your brain gets a serotonin bath, and that's really important it helps to think of it as resetting the nerves while you're asleep, and it's really important. Other you can see that our amino group becomes a little important. Gamma aminobutyric acid is an inhibitory neurotransmitter. We've got glycine, glutamate, aspartate, all these different ones. But we don't expect you to know to have these memorized. We used to. It's just too much now. Don't worry, we'll get you in college. Some little quid pro quos, things to know. Acetylcholine, we think that its levels are really important when it comes to learning and memory, and we want you to get a good sleep every night, so you reset these levels, right? We find it in reduced levels of people suffering from dementia, Alzheimer's, things like that. And it's interesting, it's an amine-based neurotransmitter, right? Norepinephrine, when your kidneys release, noradrenaline, right? As that gets around, coming from your adrenal glands in particular, that has an excitatory effect on basically neurotransmission. So, for example, I was in a car accident one time, and when my adrenaline began to surge, I remember being able to almost distinguish time as if it was going very slowly. I saw the dash coming towards me and crumpling almost in slow motion. I felt a little bit like Peter Parker, the Amazing Spider-Man, and as if I could just get out of the way. And that's exactly how your body's hardwired. It's it does a surge of these neurotransmitters, increases the signals, so it's almost like you're perceiving life normally at 30 frames a second, and on adrenaline, I felt like I was perceiving the world at like 200 frames a second. Everything, smells, heightened, everything. I can still to this day remember that whole incident as if it just happened. Yeah, I don't. it's not a. It's not a sensation you care to experience. Although some people are addicted to this feeling, jumping out of a plane, that rush of adrenaline because your senses are so heightened. The third one that Warren's mentioning is dopamine. Now, dopamine, it's really an important chemical. If you don't get enough dopamine, you can experience schizophrenic-like symptoms. Um, it's not the only reason. Sorry, my iPad went to sleep. But it's generally kind of an inhibitory sort of evening out sort of neurotransmitter, and it reduces your stress. You have more balanced walking, things like that. If you stay up super late, your brain doesn't get a dopamine bath, you'll have the shakes, and now you know why. Um, Dopamine itself, it'll make you feel tired, and if well, that makes sense. So you're getting a dopamine bath every night, but it's derived from the amino acid. That's what AA stands for down there. Tyrosine. So when you eat a lot of turkey or you drink warm milk, right, it tends to help with your dopamine production, as well as your melatonin, which helps you go to sleep, right? The fourth one, serotonin, um, when you're sleeping, you have some cells in your brain that will produce, um, uh, basically, that will produce serotonin. And think of that as your big chemical reset when you sleep. And you get some serotonin from tryptophan, which is an amino acid you find also in warm milk and turkey and things like that. But serotonin itself will make you feel majorly drowsy. After eating a big turkey meal, you'll be on the floor asleep, right? And tryptophan is also found in, well, in relation to melatonin. Our brains produce melatonin. You might have heard of it. There's a little gland in your brain called the pineal gland, sometimes represented as the third eye in some cultures. And when you're young, it's quite vibrant and it produces a lot of melatonin, and it helps you to sleep, right? And you have sort of regular serotonin baths for the brain when you sleep. That's a good thing. As you get older, the pineal gland in the center of your brain isn't producing as it used to. So one thing that we do is you can get melatonin in a pill form, but the good old milk and turkey would also help you sleep because chemically they've got tryptophan, which is the starting point of serotonin, helps you to get to sleep, right? Helps you to increase your levels. We're going to stop there because the next major installment is the peripheral nervous system. And we do talk about how our peripheral nervous system can supercharge us when we get into accidents and things like that. A few things that I just mentioned. But the peripheral nervous system is its own lesson. So that's it, folks, for this round. Thanks for stopping by. Ciao.